about the meeting. So I would like to remind everyone who hasn't done it yet to please put your name and affiliation in your username, because we need to confirm your attendance and is in accordance to the registration of the conference. Assalamualaikum warahmatullahi wabarakatuh, Bapak Ibu sekalian yang saya hormati. Selamat datang di webinar ForMIND Institute bekerja sama dengan DRPM Ristek BRIN. Dan kami mohon agar dapat mengganti nama screen-nya sesuai dengan registrasi, menyingkat afiliasi, dan juga nama Bapak dan Ibu. Sebentar lagi akan kami mulai tepat jam 7 malam, pukul 7 malam di waktu Indonesia bagian Barat. Oke, that was Indonesian language.
Now, please speak in Japanese language, please.
Then maybe only I can understand. Or maybe Pauki also, because Pauki spend your time in Japan also, right, Pauki?
4 years in Tokyo.
Oh, yes. So maybe you know Japanese words. So we're still going to wait for all of the participants to join us. And probably we will start at around the time. So Jules, I heard that your lab is one of the labs assigned for COVID testing.
I've been involved in COVID testing, but I was helping in one of the government labs.
Oh, is it your lab in Imperial or in Cambridge?
No, it was neither actually. So it was a government laboratory for, I don't know if you come across UK Biobank, it's a large scale epidemiology study, but they set up a mega lab there where you could have, I don't know, did we have about 40 people per shift in their testing? And the aim was to, to get through tens of thousands of samples per day.
Very large amount of testing there.
Yes, yeah.
But you're doing the volunteer like yourself, or your staff is doing the testing?
No, I did it myself. So I was in one of the biosafety cabinets. Yes, yeah.
That's amazing.
It was pretty tiring. Yeah, I think it's the longest I've been in the lab in quite a while.
Yes, I can imagine. Maybe it's, this is like duty calls, right?
Exactly, yes, yeah.
So the situation now is getting better? The labs are starting to reopen little by little, period?
Yes, yeah, so we've been opening, we opened up the labs in June. We're under certain restrictions at the moment. And what's it like in Indonesia?
Many of the labs are still closed. The university is still closed, and learning is done online. And I think there's about to start opening the laboratories maybe little by little, but still very much restricted asset. If I'm not mistaken, yeah, Pauki.
Yeah, actually.
So we're exactly at 7 p.m. now. So for admin, perhaps you could display the screen. Okay, we're about to go live soon. And yes, I will just wait for the admin to display the screen, the welcome screen, to display again the rule of the meeting today. Marvin, sudah bisa di-share slide-nya atau Sri?
Ada di Mbak Sri, Bu.
Mbak Sri bisa segera di-share?
Iya, Ibu.
Yes, Assalamualaikum warahmatullahi wabarakatuh. Sebelum kami mulai, mohon ke slide sebelumnya Mbak Sri. Kami ingin mengingatkan sekali lagi beberapa peraturan yang terkait dengan penyelenggaraan acara webinar hari ini. Mohon slide sebelumnya. Yang pertama adalah kami akan mute speaker Bapak dan Ibu selama acara berlangsung, dan akan kami buka speakernya di sesi diskusi tanya jawab. Jadi mohon untuk tidak menyalakan audio ketika acara berlangsung untuk kelancaran dan kejernihan suara. Lalu nanti akan dipersilahkan untuk bertanya. So you're allowed to ask questions during the talk. by posting the questions using the message to all participants. Dan nanti kami akan memilih pertanyaan-pertanyaan dari Bapak Ibu yang ada di kolom chat dan bisa mulai bertanya setelah presentasi dimulai. Lalu nanti setelah presentasi kami akan tampilkan pertanyaan yang terpilih untuk nanti kemudian dijawab oleh narasumber. Demikian. Dan sedikit saya ingin menyampaikan tentang rundown acara. Yang pertama akan kami mulai dengan pembukaan dan juga perkenalan dari Pak Oki Kanarajasa, dari Prof. Oki Kanarajasa, Direktur dari Research and Community Service, Ministry of Research and Technology. And then followed by scientific writing lecture dari Professor Julian Griffin. Lalu followed by Q&A and then closing. So I would like to welcome everyone to today's webinar. And I really would like to deeply convey my gratitude for the attendance of Professor Oki Kanarajasa and also our speaker today, Professor Julian Griffin from Imperial College London. And I would like to invite Prof. Oki now to please give his opening remarks.
Okay, thank you very much, Dr. Sastia. Good morning, Jules, and good evening, everybody. Welcome to the second series of online seminar by world-class scientists. And please allow me to deliver a short opening remark regarding the today's session on scientific writing. So my title of remarks is Scientific Publication, an Assessment of Research Productivity and Institution Reputation. I'm the Director of Research and Community Service, the biggest funding agency for research and community empowerment for Indonesian lecturers. Let me allow to start with the fact about the resources of Indonesian lecturers. So basically we have around 290,000 lecturers distributed in about 4,700 universities. But there are only about 3,250 universities under the Ministry of Research and Technology, but still, we are still the biggest university in the world. And we have also 5.5 million undergraduate students, 326,000 master students, and 33,800 doctoral students. So basically, if we are looking at this modality, so actually Indonesia has a very good for producing a good papers as a result of dissemination of our research results. And when we talk about the targeted research, especially in the university, then normally we'll talk about the three different things. Number one is research productivity. For example, how many papers published in international peer-reviewed journal, how many competitive research grant obtained by the staff or lecturers, and number three, for example, also the numbers of intellectual property rights. And then when we move into the next level, then we talk about research impact. For example, how many citations of the paper produced by Indonesian scientists, and how many research-based teaching material, and how many research-based teaching book as well as outreach. And for the next level, which is research excellence, it is normally indicated by the highly cited papers, the H index, and the publication in highly impact journal, and number of center of excellence. That's the ideal situation. And when we look at the role of publication in the World University ranking, basically we will see that there are three different ranking system in the world. Number one is academic ranking of World University, or Shanghai Jiaotong criteria, and then follow the Time Higher Education ranking, and number three is QS World University ranking. Please take a look that for each different ranking system, they include publication as a part of the indicator for the overall grade for the ranking. For example, in ARWU, they indicate using high citation of the researcher that's responsible for 20% of the total grade, and for the time higher education, even higher, 32.5%, and for QS is about 20%. Of course, at the moment to be realized, we cannot use ARWU as a reference for most Indonesian university because of very high level or criteria. For example, they measure about how many nature or article published in science and how many staff receive Nobel Prize or how many alumni they receive the Nobel Prize as well. So at the moment, Indonesia will think about at least time higher education or QS ranking. So that is why I relate that research productivity is very much related to uh, institution reputation. This is, for example, the criteria for the time higher education. As I indicated using the red circle, there is one aspect which is citation per staff, 20% of the total evaluation component. And it is indicated that it will be based on 5-year publication data with citation from Scopus. How the ministry consider the publication, especially international reputable publication, as one of the key performance indicators in research and innovation. As you can see here, number one is the number of registered IPRs, but number two is international publication. The target in 2015, only 5,000 papers, and 2019 is 19,000. Please consider that we have almost 300,000 lecturers in university, plus about 12,000 researchers in the non-university research institution. And this is the performance of the Indonesian scientists. On the above, you can see from left to the right, the target in 2015 only 5,000, but the achievement was 8,000 something. And in 2019, the target was 19,000 and achievement was 46,000. So you can see just within less than 5 years, we reached very significant increase of the number of the international publication. Even in the below graph, you can see among selected ASEAN countries, within the last 2 years, Indonesia also achieved the highest ranking. The concern is that in 2015 until 2019, overall of the publication, 47% were conference proceedings indexed by Scopus. But this is the fact because we have a very huge disparities in terms of the quality of university between Java and non-Java island. This is the last data on the publication between January to June 2020. Yes, we still number one. In total, we reached 19,000, but still about 37% were conference proceeding. But this is a good bit improvement because last year the proceeding was 47%, now become 37%. So the challenge is how to motivate those who published previously in conference proceeding or low-level Scopus-indexed journal to migrate into higher level of publication. The challenge is we need capacity building in terms of how to write good scientific papers. Publication also very important in terms of clustering Indonesian university based on research performances. We have conducted this every 3 years. And as you can see here, for the component evaluation of research output, that's responsible for the 50% of the total grade, mostly based on research performances, which is publication. So publication will determine the cluster of overall Indonesian university. They are classified according to different clusters, just like your credit card, Jules. We have platinum, we have gold, silver, bronze clusters. So this has two consequences. Number one is the budget quota, that platinum will receive the highest and bronze will receive the lowest. In addition, for platinum, gold, and silver, they also entitled to manage what we call it decentralized research budget. And also, in terms of assessment of the proposal, last year we received 22,000 research proposals, and we will assess their proposal based on three different aspects. Number one is track record, and this is mostly also dominated by publication, and then the content of the proposal as well as the budget. So we can see here that there is a strong link between research productivity in terms of publication with institutional reputation in terms of clustering or their ability to secure external funding as well as to support their ranking in the global level. So, Jules, I would like to have your kindness to help us to share your very valuable insight and experience to prepare good scientific writing for our scientists so that they will be able to write or to migrate into a high level publication in the near future. Thank you very much for your kind attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you.
Yes, thank you very much, Prof. Oki Karna Rajasa, for your kind introduction and opening speech. We are now well informed of the status of publication as well as the mechanism on how to get grant funding in Indonesia. So before we start to proceed to the next event, which is our main event, talk by Professor Julian Griffin, I would like to invite everyone here present to have a photo session. And if you would like to be a part of the photo, please turn on your video, and then our admin staff will take your picture in the count of 3. So, okay, so we can unshare the screen now so that we have a wider view. Okay, silakan di unshare screen-nya, yeah. And yes, on the count of 3. Admin, sudah siap untuk foto? Okay, so in the count of 3, I would like to take a picture for everyone. 1, 2, 3. Okay, one more, please. Maybe with a thumbs up. 1, 2, 3. Okay, thank you very much. Admin, is this already okay?
Yes, okay.
Okay, thank you very much. Okay, now we would like to proceed by thanking again Professor Oki Karna Rajasa for your opening remarks. And I would like to introduce our main speaker for today, Professor Julian Griffin from University of Cambridge, and now he's in the Imperial College London, just moved to a different position. And I know Jules for quite some time, Jules, through our involvement as fellow board of directors, in which he acts also as the president for the Metabolomics Society. So it's been a really great honor to be working alongside him for the development of metabolomics research field. And his expertise is on the metabolomics and lipidomics tools to investigate aspects of metabolite syndrome. And In addition, he is also a recipient of the Thought Leader Award in Mass Spectrometry from Agilent Corporation, and many other active involvements in key organizations in metabolomics research field. And he has published more than 300 publications with an H-index of 68. So very experienced and seasoned researcher for scientific writing. So we are very honored to welcome Jules today as our speaker to deliver his talk on scientific writing. So Jules, time is yours, and I would like to ask you to please start your presentation.
Okay, well, thank Professor Oki Rajaza for the very kind invitation as well, and the introduction, and the bigger picture actually about where this all fits into as well. So hopefully you can see my slides coming up.
Yes.
Just got to put us into, there we go. So today I was going to give you an introduction to scientific writing, and I'll tell you more about the scientific writing in a moment. What I'm going to do is just allow me so I can see a bit more of my screen. But Saskia said that it might be useful to start off with a little introduction into what my group does in terms of research, so you get a feeling for some of the wider research interests that we have. So as Saskia's already mentioned, I spent quite a lot of my time in Cambridge, at the University of Cambridge, in the Department of Biochemistry, but also part of the Medical Research Council's unit at Human Nutrition Research, also in Cambridge. So about 18 months ago, I moved, I'm going to try and see if I can move this bar down. I'm always a bit worried that I'm going to end up, there we go. I'll move it. Is that better? I think that's better. About 18 months ago, I moved down to Imperial College to join the Department of Metabolism, Digestion and Reproduction. And I head up the section of biomolecular medicine. And really the aim of this move was to move some of the things that we've been developing in the lab into the clinic to get a better understanding of what's happening in terms of human health. So I can... My group is interested in type 2 diabetes and the metabolic syndrome. So if you're not familiar with the metabolic syndrome, this is a cluster of disorders that are associated with obesity and also the increased risk of developing type 2 diabetes. As I'm sure many of you are aware of, we're currently in a type 2 diabetes epidemic. So when we come out of the COVID-19 pandemic, many of us will be returning to sort of trying to understand why so many people are developing type 2 diabetes. But it's really only sort of part of the story in terms of what's happening in terms of metabolic health in individuals. And we have this cluster of disorders called the metabolic syndrome that seem to interact with each other and increase the relative risk of developing these diseases. So if I can, I might try and do here. Hopefully you can see the laser pointer now. What you can see across the world here, so this is prevalence estimates for 2025. You can see that everywhere is predicted to have increases in the incidence of type 2 diabetes, but we can see that certain countries are going to be much more affected than others. So in particular, in the Middle East, we're going to see a big increase, but also places like Mexico and Brazil in terms of that prevalence of both type 2 diabetes and the related disorders that are associated with this. Surprisingly, at least for my research, is that pharmaceutical companies aren't targeting their therapeutics to type 2 diabetes per se, in that we have pretty good generic compounds on the market for treating type 2 diabetes, but really they're aiming at some of the associated diseases that are associated with the metabolic syndrome and also type 2 diabetes. So looking at fatty liver disease, kidney disease and heart failure going into the future. So my group's aim is to understand why certain individuals develop insulin resistance and then type 2 diabetes while others are protected. And in particular, I've had a long-standing interest in how diet interacts with this risk. Now, what we found is that we need to do this at the individual level. So using metabolomics and other omic approaches for so-called deep phenotyping. The aim here is to try and cover as much of the metabolome or other omes, the transcriptome and the proteome, to get a big picture of what's happening at that individual level in terms of the disease process. Now, this is great if you're studying a mouse model of the disease or a rare clinical form of the disease, but it's not very useful for telling us about why at a global level we might be seeing increased prevalence of type 2 diabetes. So we've also been developing tools for doing what I describe here as simple, robust assays, for doing things at the epidemiology level. So can we do this type of analysis in thousands, if not tens of thousands of samples? And I'll show you some examples of where we've applied that in a moment. So you might think that actually we know all there is to know anyway about how nutrition interacts with our risk of developing type 2 diabetes and obesity. This is a relatively recent meta-analysis that got a lot of people talking about the interactions of diet and cardiovascular disease. So many of us will know innately that increased diets where we increase saturated fat intake should increase our relative risk of developing cardiovascular disease, atherosclerosis, and also suspected type 2 diabetes. But this is still one of the largest meta-analyses to date. And I just draw your attention to the conclusion from this meta-analysis, that combining all of the previous large cohort studies where people had measured fatty acids using approaches such as gas chromatography mass spectrometry, and then looking at associations with cardiovascular disease, the authors concluded that current evidence does not clearly support cardiovascular guidelines that encourage high consumption of polyunsaturated fatty acids and low consumption of total saturated fat. So this is quite shocking. And really the problem that we're faced with is actually we haven't been measuring these fatty acid changes in the diet and indeed in blood plasma very well. We're using GCMS, which is a relatively crude approach in terms of doing this. It has to cleave those fatty acids off the lipid that they're actually associated with, so we lose information during that process. And in some of these older studies, the data is quite noisy, both being an older study because what other components in the diet, and also just the analytical challenge of trying to do this in tens of thousands, if not hundreds of thousands of individuals overall. So we've been trying to develop assays where we can do this type of analysis for thousands of samples. So far we've used it to look at 3 different cohorts. A cohort called Finland, which is found in the Cambridgeshire area, based around Cambridge and the city of Ely. If you know your UK geography, it's on the eastern side of England. And this is led by Nick Wareham and Nita Farui. The idea behind Finland was to try and understand why people develop type 2 diabetes. And this shows you a summary of these 1,500 individuals that we've measured. So each one of these green dots represents a blood plasma profile as measured by mass spectrometry. And then right in the middle are our QCs. These are pooled QCs that we've run across these 1,500 samples. And this is a type of analysis called principal component analysis. And what you can see is that our QCs very nicely sort of line up in the middle of this analysis over here. And we've got 1,500 individuals across here. So this means that we've got a nice robust assay in terms of doing this type of number of analysis. We've since moved on to sort of larger scale studies. So Promise is based in Pakistan. This is headed up by John Dinesh at the University of Cambridge. And we've looked at 5,400 individuals from Promise and looked at their relative risk of developing myocardial infarction using this approach. We're currently just completed around about 30,000 samples in another study called Interval, and we're hoping to bid for UK Biobank, which is 500,000 people in total. Just down here shows you sort of what we're aiming to do down here. So this is another type of pattern recognition approach down here called orthogonal partial least squares discriminant analysis. And hopefully what you can see is that we've been able to separate out the healthy individuals, in this case in the Finland cohort, they're found in green, from those that have fatty liver, so simple steatosis that formed in these individuals. Now, this is a useful plot in terms of diagnosis. You probably wouldn't rely on it in terms of its robustness, but what this allows us to do as biochemists is to begin to understand what metabolic pathways are driving these individuals into developing fatty liver disease, and why these people over here in green are protected from fatty liver disease. And without going into too much of the details, this is a hierarchical cluster analysis down here of all of the triglycerides that we've detected in this data set from Finland. And what you'll notice here is that we've got 3 distinct clusters over here of triglycerides. So one down here, one here, and one up here. So these triglycerides are behaving differently. And in particular, it's a triglyceride cluster which contains more saturated fat, so fats such as palmitate and stearate seem to be driving this separation between fatty liver disease and healthy individuals. And we think this is from de novo lipogenesis driven by carbohydrate and free sugars that are found in the diet. So these types of studies also lend themselves to GWAS based approaches. So as soon as you're beginning to measure thousands of individuals, you can begin to look at the associations, in this case between lipids that we've measured by mass spectrometry, and then SNPs that are associated with diseases that can be measured by microarray or next-gen sequencing. So this shows you some results here from the PROMIS cohort, where we've looked at people from Pakistan and looked at individual lipid species that we can detect. So at the top, we're looking at cholesterol here and seeing which SNPs are most associated with cholesterol. So no surprise, we get LPL, ApoA5 and ApoE, all known risk factors for cardiovascular disease. But we can also begin to look at some of our triglycerides, which we know are associated with fatty liver disease, and begin to pull out SNPs associated with the disease over there, and begin to sort of pull out some novel SNP associations as well. To show you where this is all going, this then allows us to build up a metabolic network of how lipid metabolism interacts with your genome. So in this case, we've got a Gaussian graphical network model here of SNPs known to be associated with either dyslipidemia or cardiovascular disease. They're found in these pink ovals over here. So you can see XBP1 over here, PNPLA3, a lot of interest in PLA3 recently from fatty liver disease. But we can also look at their associations with key lipid species that are found in these blue boxes over here. So this is free fatty acids, lysophosphatidylcholines, cholesterol and derivatives, and so on and so forth, and begin to build up profiles associated with these disease processes. So that shows you one approach where we do things at scale. We've also got an interest in doing mass spectrometry imaging. So this is on a much smaller scale in terms of number of individuals or number of animals that we can see. But in this approach, we can take tissue samples and analyze them directly by mass spectrometry. So we spray a matrix onto the tissue sample and then raster a laser across the tissue section and generate mass spectra associated with each one of these pixels as we go across the tissue slice. So we've used this, and this is work conducted by Zoe Hall, who's now a lecturer at Imperial College. We've used it to look at the development of fatty liver disease, in this case in an animal model in the obi mouse, which eats itself into, first of all, type 2 first of all into obesity and then into type 2 diabetes. And we can look at the relative changes in lipids as we go from healthy individuals into, in this case, mice with steatosis with inflammation. And what we find in the liver is that liver consists of a number of zones in terms of its metabolism. And the mass spectrometry imaging nicely shows that we get this loss of zonation as the disease progresses. And it's in part driven by phosphatidylcholines, which can contain arachidonic acid. We think that's driving inflammation in these mice, and indeed in people that suffer from non-alcoholic fatty liver disease as well. Just as a little aside, we also took part in a study relatively recently called Extreme Everest 2. So the study itself took place in 2013. It took us a while to publish the results from here, and some of this work is still ongoing. But this is a much smaller study here, comparing Westerners and how Westerners, largely people from the UK, adapted to high altitude by taking people up to base camp at 5,100 meters above sea level. So this is a group of volunteers here, and you can see down here I'm wearing a fox hat over here, and this is me at base camp, and then this is me up here with one of my then PhD students at the testing facility. But we were interested in comparing Westerners and how they adapted to high altitude compared to Sherpas. So one of the native populations in Nepal, these Sherpas had lived at Kathmandu all of their life. But we know that they're genetically better adapted to high altitude. So we measured how they adapted to high altitude. To give you a little bit of the background, we know that Sherpas have a number of SNPs in their genome which are associated with this high altitude adaptation. And in particular, there are SNPs associated with a gene called PCr alpha, peroxisome proliferator activated receptor alpha, which is an important transcription factor, turning on fatty acid oxidation, regulating gluconeogenesis and fatty acid synthesis as well. So we used our metabolomics tools to see how people adapted to high altitude, and in muscle biopsies that we took from both cohorts, we were able to look at how the TCA cycle or Krebs cycle adapts to high altitude and find very different responses in the Westerners compared to the Sherpas. At the same time, at base camp, we were also able to do oxygen electrode experiments to look at mitochondrial function in both groups, and again found striking differences between the Sherpas and the Westerners that we were comparing. And the final slide that I just wanted to show you is just some of the results that shows just sort of how different the Westerners were in terms of their adaptation. So this graph over here is a mass spectrometry measure of phosphocreatine. So this is one of the main buffers for phosphate inside the muscle for maintaining ATP. So here we have muscle biopsies from Westerners at low level. So this is at the initial assessment in London. And this is when they arrive at base camp. And this is after 2 weeks at base camp. We get this profound decrease in phosphocreatine in these individuals. We look at the Sherpas. Sherpas at baseline, which is measured in Kathmandu, and then Sherpas when they arrive at base camp. And you see this significant rise in phosphocreatine, so a very different physiological response. So hopefully that's given you an idea about how we're using metabolomics and lipidomics to look at disease processes, but also physiology at the population level, but also at the individual level in order to understand these disease processes. So, like I say, hopefully that's given you a bit of a background idea to my own sort of research interests, and hopefully some of those research interests are also an interest to the people in the audience. However, I've been invited today to tell you about writing papers. So I felt I needed to justify what my experience is in terms of writing papers. Saskia very nicely dropped in my H index sort of earlier on. But really I'm here as a member of the editorial board of 3 different journals. So I sit on the editorial board for genome biology, scientific reports, and metabolites. I think, you know, it's fair to say that I do a lot more for scientific reports and metabolites, probably more so for metabolites, than I do for genome biology. And that's because it's run by a permanent staff of editorial board members, a lot more than relying on its academic editorial board. This is not me, it's just how it feels when you're having to review a lot of papers. And as a little thank you and a little disclaimer, some of these slides are from Professor Roy Goodacre. So Roy is the editor-in-chief of the journal Metabolomics. and he very kindly provided some slides to show you how the review process works for the Journal of Metabolomics. Metabolomics is a Springer Nature journal, and really the whole pipeline would be fairly typical for a lot of the Springer papers. And I can go into it later. Nature and the high impacts of Nature publications have quite a different handling process, as you might have found if you've submitted to them in the past. I was sort of thinking about where to start really, and before you actually even start the paper, I think my big message is it's never too early in the process to be collecting the data together. So what I find, at least in my lab, and from my own personal experience, is that papers tend to be written at the end of a studentship or a grant. But this, for a number of reasons, is pretty poor timing in a lot of cases. So it's not uncommon these days, particularly if you're going for high impact journals, that you're asked to do further experiments. So the problem with doing your papers at the end of a studentship or the end of a grant is it's at the end of a studentship, you've probably just left the lab and you don't really want to go back into the lab because you've got your thesis to write. So if you can be thinking about publishing these papers, or at least getting a draft together in your second or third year, so in the UK, PhD studentships tend to be 3 to 4 years overall. I think that would be very useful in that you can begin to think about some of these further experiments. And if a reviewer asks for a further experiment, you might be in a position to be able to add to that. I think the other big advantage is if you're writing as you go along, you can ensure your data is properly processed. And this can take your experiments into new and interesting directions. So I've often found going down the classical route of writing the paper at the end of a studentship or the end of a grant is you come up with a much more interesting hypothesis than the one that you were testing originally when you applied for the studentship or when you got the grant awarded. So sometimes it's very useful to actually be able to move in new directions in terms of these experiments. And I think the whole process of beginning to write this down, if not in a full-blown paper, at least as a report or as a talk in terms of this, encourages you to keep your data together and update your methods as you go along. So it's not uncommon when you come to write a paper that you might have a fair amount of searching to do in terms of finding where all the data has gone. And often you forget why you might have changed something in a method. So many of us will develop a method during a paper. We'll have to sort of try out things, in my case, different solvents for solvent extraction, perhaps different methods for the mass spectrometry. And it's very easy to lose some of the results or some of the reasons why during that process. And then I think what we're going to see is electronic notebooks becoming much more useful as we go on. So at the moment, I'm moving my lab, and it'd be nice to think sort of once the lab is completely up and running at Imperial College, we'll all be using iPads or something similar rather than the classic lab book that we have done in the past in terms of keeping all this data together. So let's imagine that you're at the situation though where you feel like you've actually got enough of your results together and you're ready to write a paper. So people have a number of different ways of writing papers, and I think my message here is whatever you feel most efficient with. So there's a question about whether you write figures first or do the introduction first or start with the results section first. So people or groups of people have different approaches to writing a paper, and I've tried both going in the classical direction where you'd write the introduction first, go through the methods, results and discussion. I've also started where I've started writing the results section, and I've also started where we start off with doing the figures and then we write the rest of the paper around the figures. And what I'd say is that all three methods work equally well or equally badly. There'll always be a crunch point, I think, where you might need some help getting over the activation barrier to write certain sections in those papers. And there's no one size fits all. And really, it's whatever works for you or the collaboration of authors. So like I say, you don't have to write the paper linearly, so going from the introduction through to the discussion. And indeed, one of the problems with writing the paper linearly is that typically people would write the introduction first, then the methods, then the results, finally the discussion. We spend a lot of time on those sections. The thing that a lot of people forget is the first thing that the editor is going to read is the abstract. So a big problem with writing it linearly and not starting, at least in this case, with the abstract is that the abstract often misses key information and it can sound quite bland if it lacks that information. It just ends up describing the study that you did and not so much the results that you've achieved. So have a think about sort of focusing on the abstract, making sure that the abstract has enough time when you go about publishing it. As I've already said, some will start with figures and then build the paper about it. So I'm part of a large-scale sort of epidemiology study at the moment, and that was the route that we went down. So we initially started off with figures, produced some very nice figures. The sad thing is that some of those figures have had to go since then, and we've had to sort of adapt around it. It's often easier to write the methods, and if you're writing a thesis, this is often the first chapter that people will write up as part of this. I think a key message, though, for everybody is that when you write your first paper, try to get input from all of the authors. So everybody should contribute towards a paper, and as an editor, this is really key. So many manuscripts now require a section on authors' contributions. So what have you contributed to that particular paper? It's useful to get wide input, especially if it's your first paper. So try and go out of your way to make sure that all of the authors have the opportunity to contribute, and no matter how important they are, that they do contribute to the paper, and it will make for a much better paper. For high-profile journals, it's good to get input from people who regularly publish there and from scientists outside your own research area. So if you're going to try and publish in Nature, and I'll lay my cards on the table, I find, I've got one paper in Nature, and that was a complete surprise that it got into Nature at the time. So I don't have any sort of tried and tested route. But for any of these high impact journals, try and get a scientist that's in a related area, but not too closely related to your research. So for me, involved in metabolomics, I would try and find one of my biochemistry colleagues or one of my medical colleagues that doesn't study that particular disease process, to see whether they find that the paper is interesting and accessible as well. And also a native English speaker can be very useful for papers. So I'm very fortunate in that it's been decided over a number of years that English is going to be used as the lingua franca of science going forward. I'm very relieved it wasn't German, because I think that was nearly the other choice in chemistry. But it's key to actually making sure that your paper is easy to read. And in the future, we'll also be making sure that it's easy to read by a computer as well. So it's important that the English makes sense and that it's structured correctly. So some journals will offer this as a service, but it's probably not as good as a friendly colleague who will explain why some things don't sound right. And as a native English speaker, I don't understand sometimes why some things don't sound right. I just know that they don't sound right and can correct the English. So it's having somebody like that that can go through and ask you, did you really mean this, or did you mean this, as part of that paper? At the start, it's worth checking the journal's format. So each journal will have its own format. So a lot of journals now have methods at the end of the paper, or even those methods online. So for a lot of the Nature journals, they now go for STAR methods, which are very much abbreviated, and then the main section of methods are found online and not in the paper itself. Very frustrating when you read the paper if you want to know what somebody's done, but they're doing that sort of to keep the word count down in the main article. You can check on the journal's website to see what the preferred format is. And it's also worth checking a publication or two to see that the format in action. So sometimes the way they describe it is not really the way it sort of translates into papers. So it's worth having a look at some of the papers that the journal has published previously, perhaps picking something that's in a similar area to yours and seeing how you could modify that format overall. Some journals even provide a manuscript template. So analytical chemistry, you can download a Word document and then populate that as you go along. And I find that quite frustrating as a reviewer because it shrinks down the font quite a bit, so it makes it more difficult to see some of those, some of the figures and things. But you know, you as an author, you can see sort of how the paper is then laid out. So it's worth having a look what's on the journal. A few journals now allow you to have it your way. So a couple of publishers are sort of led on this. They tend to be those that are associated with some of the archive, things like bioRxiv and medRxiv, aren't too worried about the initial format of the paper when you submit it. It's only once they've agreed that to publish in the paper that they then want you to put it into the journal's format afterwards. So that's very useful if you're shopping around to find the right journal for you. Probably the biggest difference in format is references. So many of you will already discovered this, but it's an excellent idea to have a good reference manager system, especially if you're resubmitting a manuscript. So there's nothing worse than having to go through a manuscript and having to renumber everything or changing over the format of that manuscript manually. It's very easy to lose papers during that process or end up with faulty referencing. So I would recommend going to a good reference manager, and there's a number of those that are available that freely interact with Word or easily interact with Word as well. I wanted to say something about bioRxiv and other repositories. So I'm relatively sort of come new to bioRxiv. I know a number of my colleagues had liked bioRxiv and medRxiv and the other archives that you can have access to, particularly for grant applications. So at least in the UK, if you wanted to demonstrate that you had a paper as a draft, you'd put this on bioRxiv and you get a reference then, a DOI number for that paper, and then you can quote that in the grant directly. So we're not allowed to quote papers in process or papers in progress in our grant applications. We are allowed to state a DOI from bioRxiv, so this is becoming increasingly popular. It's a great way to get your research out early and make your mark. So if you're in a particularly fast-moving area of research, you get those papers out and get some feedback from readers. So we found it useful for gauging interest in terms of readers. But the other thing for pointing out is you can submit your papers directly to certain journals with little or no formatting. So there's a number of different publishers which have partnered with bioRxiv and Cold Spring Harbor, the people behind bioRxiv, that will accept those papers via a little portal there, so with relatively little extra information in terms of passing that paper on. So I thought I'd show you sort of how the review process works, and this is a slide that's courtesy of Roy Goodacre. So he's the editor-in-chief of Metabolomics. So this is the review process as it happens for the journal Metabolomics. But as I've already said, it also is a review process for a number of other similar journals out there, not just for Springer journals, but also for a wide range of sort of classical journal publishing. So first step is the submission is received by the office, and it's then assigned an editor. So not all papers will go to the editor-in-chief. Some will go to the associate editors or part of the editorial board. The editor has to consider whether it's worthy of consideration. So this is the thumbs up or thumbs down. And often this is a quick scan through in terms of the abstract and a read through the introduction and the results overall. In some cases, this reject and inform the authors, and invariably in a journal like Metabolomics, This is because either the paper isn't up to quality, so usually there's problems with the written English in that paper, or more commonly, it's outside the scope of metabolomics. So metabolomics has a scope centered around the field of metabolomics, and sometimes we receive papers which are quite far from what metabolomics involves. So the editor would reject that and it wouldn't be sent out to review. If the editor considers that it's worthy, we'd then send it out to 2 or possibly 3 reviewers overall. The 3 reviewers might be necessary in there might be some specialist reviewing required. So if there's a lot of statistics that's found in the paper, the editor might decide that they need statistical review in addition to the other 2 reviewers. The reviews then come in, they're collated, and a decision is made on the basis of those reviews. And it can be sent backwards and forwards, and it can take a while to get those 2 or 3 reviewers. I think Roy's plea was that if you're asked to review, if you can at least sort of let the editor know as quickly as possible whether you can review or not, that helps their job immensely, so they're not just hanging on for weeks waiting for you to reply. So in terms of the decisions that can be made from these collected reviews, you can get accept, revise, and revise can be minor or major responses, or reject. So that shows you reject there, and you can see the little unhappy face down here. And then we've got, if I can move, requests for revisions to be made. And then once the revision is made, that gets sent back for editorial assessment. So that goes back to the editor to have a look at. And then the editor needs to make a choice about whether it gets sent back to the reviewers or in some cases, it can be accepted at this stage. So just to go through this stage, revisions are made by the authors, it goes to the editorial assessment. If it's a major revision, some will go back to the reviewers and they'll go through another round of review process. But in a number of cases, the editor feels often that they have enough information to be able to make up a decision on the basis of those review processes, particularly relevant for minor reviews, and it can go through either back for another round of minor reviews, or at that stage can be accepted and you have the paper published. So hopefully that's given you an idea about what happens with the paper. And in this case, for metabolomics, it's published online almost straight away in terms of a preprint. I think the thing to say for somebody that's been in publishing scientific papers for a number of years now, so I got my PhD back in 1997, what I found is that actually the rate of publication or the time taken for publication has greatly speeded up across all of the journals. So it always used to be the case that the high-ranking journals were quick, but now some of the lower impact journals are just as quick these days. So one question you might have is what happens during the office checks? So right at the start of this process, if I go back here, you can see that very first step is that the submission is received by the office, and they do a number of checks as they go through this paper. So the papers are initially checked for formatting, and the paper complies with the rules of the journal. This includes copyright forms, so that authors have actually given over their copyright. Confirmation from the authors they are indeed authors. So we're often required to put email addresses associated with each author. So it's important to put the most up-to-date email address for those authors. Otherwise, in some cases, the paper can just sit there waiting for author approval. Increasingly, the office checks for data availability. So many journals are now committed to data availability and the open access of data. So they'll check that that data is present there. And certain conventions, for example, UPAC names have been observed in terms of those conventions. So just to go through some of those points, data availability is one of the things that they'll look for in a number of cases. So it's becoming increasingly important and may be expected by funders. You can even get a publication in its own right for making your data available. And there are a number of journals that just publish data. So the Nature Publishing Group's Scientific Data will publish data, and then Data in Brief by Elsevier will also just publish data and give you a DOI associated with that study. So even if your study didn't work, that data still might be very useful for other people. The repository that I use for my metabolomics data is a repository called Metabolites, but anybody involved in biology can deposit their data. And if you don't know about your repository, I refer you to MIBI, so the Minimum Information for Biological and Biomedical Investigations. And that's affiliated with a number of different repositories across biology overall. Many journals require an accession number to these repositories to show that you've actually deposited the data overall. I would say that there are ways around that. With Metabolites, you just need to start to deposit your data to get the accession number. So I think Metabolites has a number of studies where nobody's actually quite got round to finish submitting their data overall. Another question sort of during the overall submission process is data analysis. So some papers have a lot of statistics that are part of this, part of that paper. And it might require the editor to pick a specialist statistical reviewer or statistician reviewer for that data. In some cases, your paper might include novel data handling algorithms. It's always useful to have a name for your algorithm. And one of the questions that the editor and the reviewers will ask is, where can we get hold of this algorithm to check that it does what it says it does? So many journals will provide a facility to host data, but it could also be an open site on the university server for algorithms, places like GitHub. So I don't know if you can make out, but this is the welcome site for GitHub down here, and that's very popular with bioinformaticians to deposit their data for people to access it. And the nice thing about GitHub is I think you can access it anonymously, so a reviewer can pick this up without giving the game away that they're a reviewer for a particular paper. As a general thing, make sure that you conform with the field's reporting requirements for your data. So a big thing for us in the metabolomics field is metabolite identification. And the communities come together through the Metabolomics Society to come up with guidelines around metabolite identification. So for each of you, your field will have its own agreements in terms of how you should be reporting its data. So it's worthwhile being familiar with that and making sure that you conform to those guidelines. Otherwise, it can be a very quick way that a reviewer ends up rejecting a paper if they feel that you haven't provided the information that they need to review your paper. Excuse me. So during the submission process, we also have to check that we've complied with ethical standards statements. So this is really important for human participants and animal studies. So what we're looking for in many human studies is that the study's been performed with respect to the Helsinki Declaration for Human Studies. And often we require some sort of statement about ethical approval in a government, by a government body or the local university ethical review board. Animal studies often require a similar statement. There might need to be statements around informed consent, and also might need disclosure of potential conflicts of interest, which I think I'll come on to in the next slide. So increasingly important for a number of biological papers is that we're often required to state our conflicts of interest, particularly going to some of the US publishers going forward. So we have to declare any relationships or interests that could influence or bias the work. So this started off, I think, being largely a problem of those in drug development, and increasingly we're seeing this sort of all across the field. So it's really about awareness of real or perceived conflicts of interest and transparency. So, for example, you might declare where your research grants are coming from funding agencies, but also very important in terms of whether they're coming from industry, and there might be a perceived conflict of interest there. Honoraria for speaking at symposia, financial support for attending symposia, and so on and so forth. To help you out with these, a number of publishers now have little tools to lead you through these conflict of interests, so you can declare them as you go along during the process. So I thought I'd say a little bit about how important the editor is in this process. And the editor is really key to the overall process and how smoothly the process will work. Some editors rely more heavily on the reviewer's comments than other editors, but in every case, it's the editor that guides the papers through the overall process. So for every journal, the editor has to decide whether the paper is appropriate. And for some journals, Nature, the editorial team will assess the journal for general interest and scientific content. So unless you get past the editor and the editorial board, you won't get sent out to review. And this is very true for the high impact journals. And from bitter experience, I found that their editorial board meetings tend to be on a Friday lunchtime, often in the US. For some of the journals that I'm submitting to. So I tend to find out on a Friday night whether my paper has been rejected. I think that's to give us a weekend of cool off after they've rejected the paper. So peer review, I've got a little cartoon here about the whole peer review process. And I think it's easy to sort of fall into this trap of trying to ignore what the peer review our reviewers are trying to tell us about a paper. I've seen papers in the past where the authors really haven't wanted to take on board any criticism, and as a result haven't got their paper published or haven't got their grant funded. So I think my big bit of advice is consider what the peer reviewers are trying to tell you. You'll often have the chance to select reviewers or even deselect reviewers. So for the journal, it's very easy to check if you publish with the selected reviewers, so go outside your immediate circle. In addition, the editor has access to a list of journal reviewers, and the message from an editor is, remember, the reviewers do their job for free. So give them some sympathy in terms of their responses. In terms of responding to reviewers' comments, I wouldn't suggest sort of going down this route. This is from the film The Shining. But always, I think, courtesy goes a long way with your reviewers' comments. So thank the editor and thank the reviewers, even for difficult reviewers, in terms of doing their review process. As I've already mentioned, they do the reviewing for free. So, and nobody's getting paid, and many of the editors don't get paid for their editorial role either. Point responses help the editor and the reviewer to scan through responses, so they don't have to go through all of the paper. Remember, these are busy people, and often they don't want to read through the whole manuscript again. They just want to see what your response was and see that they're happy with your response. So when I'm going through this process as a reviewer, I'll quickly scan down to see my comments and then see what the response was. And then in my head sort of tick off whether I'm happy with that as a response or whether I think somebody has tried to sidestep my original criticism. You don't have to do every comment, but tell the reviewer why you couldn't address a suggested experiment. Explain it's the end of your PhD or there's no more money to do further experiments. And as a reviewer and as an editor, we're also seeing a number of people saying, with COVID-19, I can't get back into the lab to do more experiments. And people are trying to get around these things by doing more dry lab studies to address some of the reviewers' comments. Just to finish off, I've got some final sort of hints and tips to go through there. Less can be more in terms of papers. So shorter papers can be a lot more impactful, more widely that should be read, and easier to get a message across. So think about whether you have to be too wordy in terms of your descriptions, or whether you can reduce that word count down. To improve your papers, read more papers. So it's one of these things, I think you've got to know what you like to see in a paper in order to be able to reproduce it in your own papers. It's also very useful to review papers. You very quickly get to see as a reviewer things that don't work as well as some of the things that do work, and that can improve your paper writing overall. Open access is worth considering. It may cost more, but does increase the number of people that will read and cite your paper. So we're seeing a number of open access journals where those impact factors are going up quite dramatically, just because of the fact that anybody can access your papers. Another useful tip is to think about what your big ideas are in your paper. So a good way to organise a paper is to consider what the big idea is in that paper. You probably don't want more than 3 big ideas in a single paper. And indeed, some papers just have a single big idea that is discussed as part of that paper. It's very useful for considering what goes into the discussion and can help you sort of structure that discussion. And it can also make you for an impactful abstract. So as I already said, it's the abstract that's going to be read by the editor right at the start. So it's important to grab the editor's attention at that stage. There are many types of publications, so I've focused on research articles, but those can be broken down into shorter papers, such as brief communications and letters, as well as articles. Review articles tend to be very highly cited in many cases, and there's often a need to be invited, but you've got a good idea for a review article. You can go and approach an editor, and often editors will be only too pleased if it's a good idea to accept the review or commission the review. Letters to the editor or author's correspondence can end up in high-profile journals if you spot an important addition to a paper. I'll show you that in a moment, actually. And then there's also conference reports, perspectives and commentary pieces. So do have a think about the other types of publications. So this is just what happened. This is sort of my own personal research. About where we couldn't reproduce some work from another lab. So this paper came out in Nature Medicine back in 2002 and really got everybody's attention in terms of metabolomics. We tried to reproduce these results and couldn't get the results to reproduce because of some inconsistencies in the original paper in terms of the analysis. And we went back to the editors and got our paper also published in Nature Medicine, which was a nice result overall. So have a think about, about timely publications as well. It's always worth a careful proofread to save some red faces in papers. So this was a paper which was published in one of the Royal Society of Chemistry journals. And what you can find here in the supplementary is the senior author has written down here, Emma, please insert NMR data. Where are they? And for this compound, just make up an elemental analysis. So pretty much here admitting to a minor case of experimental fraud in this case, but that was left into the published article that went into there and could have had very serious consequences. Sometimes the worst happens. So sometimes you publish a paper and then you discover afterwards, for whatever reason, there are issues with the paper and you need to pull that paper and retract it. Message there is, I think, is to do it early. And I think in this case, despite it being a very high profile retraction, it could have been avoided by making sure that all of the authors had actually seen the data and had a chance to comment on the original paper, which in this case, the author is pretty much admitting to that they didn't do this in this case. Just to show you that retractions aren't that uncommon, this is a website that you can go to called Retraction Watch, and these are some of the retracted COVID-19 papers, and this list goes on to many papers which have been retracted, in some cases even before they've been published in terms of those publications. So hopefully that's given you a flavour and some ideas about what to do to get published. So in terms of acknowledgements, a big thank you to Roy Goodacre, the Editor-in-Chief of Metabolomics, and also Springer Nature for providing some of the slides around the overall flow process, what happens when a paper is submitted. I'd like to thank you for your attention and happy to answer any questions at this stage. And the final slide are some of the people that did some of the research at the start that I spoke about, and my funders overall. So thank you very much for your attention, and I'll be very happy to answer any questions at this stage.
Okay, thank you very much, Jules, for a wonderful, wonderful talk. So I would like to invite all of the participants to please click on this one button so that we could properly acknowledge. Yeah, thank you so much, Jules. It's really, really an interesting talk from you. And I think it illustrates on what you do and also all the process that everybody has to do. And you show it to us from the perspective of a journal editor, which is basically the key decision maker on whether or not a paper gets accepted. So next, we would like to show the selected questions from the audience that we think is most relevant to the topic. And first of all, I would like to thank all of the people who have asked questions. And the first one is asked by Professor Oki, actually. And it's about how is the trend of scientific misconduct recently? Is there any increase on the retracted papers? And also, there is a question from University of Bakri. I think the first one is about scientific misconduct. And the second question is about the questioner says that he often observed that even top authors or top scientists publish quite similar work, but in different journals and expressed in different ways. And would that be a problem? And would that be considered later on as self-plagiarism? So that would be the first question about scientific misconduct, about the trend, and what are the things that could be considered of scientific misconduct and related to the ethics. And the second question is about the publication in top journals. So many people are wondering what is actually the main characteristic of work that can be accepted in top journals. And sometimes they also experience that the major journals, like high rank and lower journals, the review process in top journals sometimes is very lengthy. So perhaps you can give an idea and what would categorize like as high rank and why some journals have a higher ranking and some journals are lower ranking. So that is quite basic about how we can rank the quality of journals. And I think the first two questions first you can answer, Jules, and later on we can continue with the rest of the questions.
Yeah, they're really good questions, actually. So my feeling is actually in terms of scientific misconduct, that has been on the rise. And there are certain fields where, I'm thinking about stem cell research went through a very bad patch where there were a number of major papers which were withdrawn and a number of high profile retractions. And the sad thing is, I think, there was also actually a very interesting set of cases around semiconductors where the person that had ended up having to retract all these papers only got discovered because he was publishing so many papers, it wasn't possible to do the research behind it, given his group size, and he had a big group as well. So there are these high profile retractions which have happened, and largely it's based around money. So it's people running expensive sort of super groups or having to maintain a certain scientific lifestyle, if you like, that they get sucked into either fabricating results or believing results that they really should be spending more time in order to test and not going with some of the first results that they might have from a potentially an underpowered study. And what I would say, though, is that we're probably also getting much better at finding out about these studies as well. So there are a number of different websites called things like Retraction Watch. Journals are much hotter on retractions. So I think there's a combination of the two things that with increased money in science, people are trying to do people are more tempted more often, but at the same time, I think we've probably got better at actually picking out these issues as well. In terms of sort of generic papers going into top journals and things, what you do see a bit of is the journals trying to coordinate sometimes publications. So if Nature or Nature Medicine, to name two of the top journals, are dealing with very similar subject area or papers in very similar subject areas, they'll often try and coordinate the publications between those two papers. So you can end up with two papers on the same subject. They should still really be looking at from separate directions or with separate amounts of evidence in there. But that does happen. In terms of self-plagiarism, I guess the worry is that it's very easy to self-plagiarize in the methods section. So many of us will develop a method and then we'll reuse that method sort of fairly consistently. So it gets difficult to sort of rewrite those things. So yeah, so you do see some of that self-plagiarism. In terms of people republishing results, though, that is a big no-no. And if you find examples of where people have redone the same analysis, so I think there were a couple of cases that came out of rather large labs in China where people had published the same results twice in different journals, and that's a serious infringement of copyright. It's a serious self-plagiarism act. And in those cases, they had to retract those papers. And I've seen it reported in journals like Nature and Science that increasingly the editorial team will have to search for this, these acts of plagiarism as well. In terms of getting published in the highest impact papers, I don't have any great wisdom into that because I find it very difficult. We had a paper published in Cell at the start of this year, and it took 4 to 5 years to get the paper into Cell. So some of it's about perseverance. It's important to try a number of different journals if you're getting knocked back. And I think for these high-ranking journals, the important thing is if you're getting sent out to review. So it's often that you'll get knocked back by the reviewers, but if the editor is finding it funny or the editorial team finding it interesting, not funny, finding it interesting, then that should sort of help you, encourage you to sort of keep on with those high impact journals. At the same time, if you exhaust those high impact journals, I think the important thing is to make sure that the paper is out there and other people can read it rather than just focus on those high impact journals. And I think one of the things to say is that some of the best scientific work has not been done in some of the biggest journals. So Hans Krebs didn't get into Nature with the Krebs cycle. So I forgot the name of the journal that he did get into. But there are lots of cases like this where people have had to publish in other publications. So Saskia, did that answer those questions?
Yes, I think it answers all of the questions. Yeah, thank you very much. I think it's quite clear on whether or not things are considered as scientific misconduct or why self-plagiarism is becoming more common. And I think the second question is on the timing of publication. So you mentioned that it's better to start writing as soon as possible. But there's another question also on, for example, if the research has been done 3 years ago, and then there are a lot of, and we cannot publish it as soon as possible for many reasons. And during the time, perhaps during the time that we couldn't publish it, then there's a lot of development also in the field. So what do you think about the timing of publication?
The timing of publications, there are certain areas of research where the timing of publications is fairly critical. So if you're doing a meta-analysis of the literature, then if you wait 3-4 years, your meta-analysis is going to become dated by further studies. So there'd be a need to re-update your analysis. But actually, in many of our cases, waiting 3-4 years, it doesn't matter so much. The paper still might have novelty. And even if the paper doesn't have novelty, sometimes it's actually very useful to confirm results that have already been published. Or even just say that you can't reproduce results that are published. So we have this thing called publishing bias, in that the papers that go into the highest impact journals are the papers that are the biggest surprise, because it's going to surprise the editor in some ways. So as a result, you know, if you've done something accidentally wrong, or even just purely by statistical chance, your results throw up a spurious result, you might end up publishing in Nature, and that would then become central dogma within that field. So it's important that if other people can't reproduce those results, they still publish even in lesser journals. So when somebody does do a meta-analysis and looks at all of that literature, you can then begin to sort of worry about what the major publication is in that field. So I think, you know, for 3-4 years, I wouldn't worry. And I've heard about theses that have gone in 20 years later. So at Imperial College, one of our most famous alumni is Brian May, who was in the band Queen. So he did a PhD in astrophysics and came back after 20 years to submit his PhD. And that was considered worthy. So I think the message is it's never too late. You might just have lost some of that immediacy and impact.
Okay, thank you very much for those excellent questions. And there's also a question about the contribution of authors. So two more questions. One is about the value of conference proceedings. Nowadays, we are encouraged more and more to publish our papers in scientific journals instead of conference proceedings. Do you still find value of conference proceedings? And the second one is also how to correctly determine, like, how significant is the contribution to warrant co-authorship in a paper?
Well, it's difficult to say. I can tell you what the UK standpoint is. So we're going through a process called REF, Research Excellence Framework, which sounds very similar to things that are happening with you guys from Oki's introduction, sort of right at the start. So with the research, with the REF exercise, we're not allowed to count any of the conference proceedings as parts of our REF. But we've actually moved from a situation where it was the first authors and, you know, the first 3 authors and the last 3 authors. Somebody's corrected me that Brian May finished after 30 years, not 20 years. So thank you for that correction. We've moved from a situation where it's not just the first few authors and the last few authors, it's now all authors that contribute in the REF process. Personally, though, in terms of interviewing new faculty, we are looking for faculty that are either first or second author on some papers, and then final author or senior author but one in terms of those publications, to show that you can manage a project. But we certainly don't expect a new lecturer to have all of their publications like that. And we're very aware that some of the papers are big projects, you know, so you end up with a name right in the middle of there, just like they do in some of the particle physics type papers that you see as well.
Okay, thank you very much. So I think nowadays there's a lot of tendency that we have to note the author contribution in a paper, right? Like some journals have already imposed that the contribution of each author has to be detailed as part of the manuscript. So I think nowadays it's very important not to just include authors who don't significantly contribute to the work.
Yes,
so I think this is one of important ethics in publishing.
Yeah. Yeah, and actually for our REF, that's considered, that's one of the ways that you justify that you're an author on there. So you have to put down what your contribution is to each paper, and it comes off the paper. So we've gone down that route as well.
Yes, okay, that's very interesting. So there's another question about, you mentioned there's a value in submitting your data into data repository. So the data repository is about like submitting your data first, right, before the actual publication. And is that fine? Because the publication is already out there. For example, there are some publications like Data in Brief, where people just submit the data first and then later on it's followed by the actual publication. Is that considered that the paper is already published and therefore you cannot publish it as a full paper?
What we tend to see more often is the other way around. So you publish the paper first, you submit your data to the repository at the same time, but that you're allowed a 1-year embargo, which is usually sort of enough time for you to get the paper published. So you can embargo your data across that time period, get your original publication published, and then produce the data brief. But having said that, you know, there are people that, for whatever reason, decide the study didn't work, but would, you know, actually having 50 control samples could be really useful for somebody. So, you know, they get the data brief then out. So it's a nice, useful tool as well.
So yeah, sometimes there's also a question about how to publish research work that is not, well, it's not actually answering the question that was originally asked, and therefore they cannot make a solid conclusion, but then the data itself is still useful. So that is one of the ways that such data availability will be useful for the scientific communities, right?
Yes, yeah.
Okay, so I think you can display the question again, perhaps considering the time, we have the final question. So how about your views about a review paper? Because sometimes also review paper is not counted as an original article. So what do you think about this?
So it's one of those things that it depends if you follow your H index and things. So the review papers can be very highly cited. I was sort of the opinion that actually primary research papers are the most important things. But then I got into reading some of the systematic reviews that people have done before, and they're incredibly useful. And they are counted in our ref systematic reviews because it's a great way of summarizing all of the studies that have occurred in the literature. So I flashed up a systematic review for saturated fat intake, and that was very useful just to see all of the cohorts that had previously been done in this area, analysed together and to form conclusions there. At the same time, if you're in a new field, what a review is very useful to do is get you to read around the field, get a feeling for what the literature is, but it also gets you noticed in that field. So I think you're more likely to pick up, you know, talks and things like the Metabolomic Society and things like that on the back of some of these review papers. So I think, you know, in terms of young academics, they can be very useful, even if you can't count them towards research exercises as well.
Okay, so I think you have answered most of the questions asked through the chat. I'm very pleased that most of the questions have been answered. I hope these are satisfactory for all of our audience. Thank you so much for excellent questions and also excellent answer by Jules. So I would like to close this by again giving you a big round of applause for your really, really wonderful talk today. And to give a token of our appreciation, Jules, we also prepare a small gift for you that we will also send through email. We prepare a caricature of yourself.
That's brilliant.
Yeah, and it's you wearing our traditional batik, which is our traditional clothing, normally handmade or printed. And this is the background is Borobudur, one of the wonders of the world. Very old Buddhist temple, and it is huge, perched on top of a hill. It has been there since the, I think, 7th century or something. So whenever, if you have a chance to visit, hopefully when the whole this pandemic finish, I hope you can take a picture with this background sometime in your visit in Indonesia. So again, we would like to close this by truly thank you for sharing your wisdom with the scientific communities in Indonesia. And we would like to have some announcement also for the next series in the World Class Scientists. Perhaps some of the participants here have registered for the 4 series. So, but for those who just joined for this particular series, we want to inform you that we will have 2 more series under this topic, and it will also be by female speakers, and they will also talk about their tips on how to manage work and life balance for women scientists. And our next speaker, who will be talking on the 30th of July, will be Professor Eriko Takano from the University of Manchester. So same region, also from the UK, similar as Jules, and the same time. And the last is also from United States, from Harvard University. Jules know her very well because she's also a board of director of the Metabolomic Society. And she will also be talking about how to establish yourself in the field, how to build good team building and network and cooperation. And especially that will be, I think, very useful to be a solid researcher in your respective field. So next, please. So I would like to also say hello for those who are now joining us from YouTube. So Jules, in addition to more than 130 people who are joining, there are also many people who are unable to join via Zoom, for example, some technical issue, and they are joining live. Almost 100 people, I think, now is joining live from YouTube. So I would like to say hello. Hello, everyone who are joining from YouTube. And I would like to ask everyone here to please visit our YouTube channel and like and subscribe so that we can continuously give useful knowledge like this and distributed it for the community at large. So I would like to please ask your help to support us. It's free. It's just by liking and comment and subscribing. Okay, so next, please. And I would like to announce that we check the consistency between your name in the registration and your name who are participants of today's webinar, and we will distribute the e-certificates for you at the latest 7 days after the event. And for those who are joining us through Zoom, you will also get access to Professor Jules' slides. Okay, next. And I would like to close this also by informing that after the world-class scientist series finish, we would also have 2 very nice events following the speakers coming from the female scientist perspective. And we will have a Girls in Science series, and we target high school students and also undergraduate students to make an essay. It's an essay competition, and it's open nationwide. And especially, we would really hope for contribution from people who are outside of Java. We really hope that this information can be disseminated to your students and your colleagues or your brother or your sister or your daughter to please encourage them to write an essay. So as a part of scientific writing, it's also very important to articulate your ideas in form of writing. This is a very good practice for our young generation in Indonesia. So we will distribute this information for all of the participants in hope that everyone will help to distribute. And you can also get this information through our Instagram account, and then please share this information. Okay, thank you very much. And the next slide, please. So I would like to say thank you once again to everyone who are in attendance. And for the last, let's open our audio and then give a big round of applause to Jules, Griffin, and to Prof. Oki Karnarajasa for giving us opening speech. Thank you very much, everyone.
Thank you.
Thank you.
Thank you so much. I hope you all enjoyed the talk, and we'll see you on the next WCS series of ForMIND Institute. Thank you very much, Jul. Wassalamualaikum warahmatullahi wabarakatuh semuanya di Indonesia.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam.
Terima
Kasih banyak.
Terima kasih.
Terima kasih semuanya.
Terima kasih
semuanya.
Terima kasih.
Terima kasih Pak Oki.
Terima kasih Pak Oki.
Terima kasih Pak Prof Oki.
Terima kasih.
Matur nuwun.
Prof Griffin.
Sampai jumpa besok pagi, Prof Oki.
Yeah.
Prof Oki. Thank you.
So we'll see you on July 30th, hopefully. Bye-bye.
Bye-bye.
Bye.
Bye.
Dadah. Tutup.
Done.
Sticknya hilang. Nah, dah. Udah. Iya. Oh, udah nggak usah ikut, masa laki-laki mau ikutan.
Thanks very much, everyone. I'm going to dash off. I've got to get ready for another meeting, but it was great, actually. I enjoyed it.
Yeah. Thank you so much, Jules. Thank you very much.
Thank you very much for the invitation. I enjoyed it. Thank you.
Thank you.
Good night.
Yeah.
Bye.
Thank you.
Alhamdulillah.